Hi, I think I know everybody. Bill will speak later about MS-2 and PhiX-174, but I will give you just a brief overview about virus. So, what is a virus? By definition, virus is a potentially pathogenic infectious nucleoprotein entities that present only one type of nucleic acid, which is reproduced from its genetic material. Basically, it's a cellular organism, so virus has no cells. It's a mandatory intracellular parasite which not replicate individually, which means that virus needs a cell, a host, to reproduce. It's the most abundant entity on the planet. We can find virus everywhere. Virus have the ability to infect all life forms, including virus. So we have virus parasiting animals, plants, and bacteria, and virus also. It is considered the main agent of evolution because viruses have the ability to function as horizontal gene transfer vehicles. So we have basically three domains. We have, sorry, the figure is in Portuguese, but here we have bacteria, here we have archaea, and here we have eukaryotes. So this is the last universal common ancestor, so it's the first cell ever, and which originates three domains. And here we can find virus. The biology, here we have different kinds of virus. So I think you're familiar to HIV, Ebola, and influenza, and all kinds of virus. We have two distinctions among them, basically. So the virus which infects eukaryotes, which means the virus which infects humans and animals, and it's called virus. But the virus which infects virus and bacteria are called bacteriophages. And we are focused on it because it's what we are using in the SafeWater project. So here, basically, we have two kinds of virus. These viruses infect humans. So we have one without the envelope and one with the envelope. This envelope is a bilayer formed basically by glycoproteins. And here we just have the capsid that protects the nucleic acid, okay? And here we have the bacteriophage, which is the virus which infects bacteria and another virus. And specifically we have MS-2 and Phi, which infects E. coli. So I will just show a video to show you how these bacteriophages infect the E. coli, infect the cells, and reproduce itself.
Yeah.
So here we have E. coli. And here the bacteriophage. So the virus is infecting the cell and puts the genetic material inside the cell. The material is replicating, and at some point the cell bursts to release new virus. So we have a cycle, and this virus will infect another cells to reproduce more and more and more. So lots of E. coli, lots of bacteriophagy, and we have an infection, okay? This is the complete cycle. So Bill will speak more about it, so I'll be brief. The difference between MS-2 and Phi is that MS-2 is a single-strand RNA virus, and Phi it's a single-strand DNA virus. Except for one kind of virus, they only have one genetic material. So here we just have RNA, and here we just have DNA. And this virus, this bacteriophage, is resistant to chlorine, and this bacteriophage is resistant to UV light. Sorry, the opposite. Sorry. So why we are using MS-2 and Phi? Because our goal is to provide clean water, right, for everybody, and we use basically chlorination and UV light to disinfect the water. So according to EPA, there's an hierarchy, a viral hierarchy. So we have small and non-enveloped virus that are most difficult to inactivate, large and non-enveloped virus that are moderate, and enveloped virus that are easy to inactivate. And we can find MS-2 and Phi in this category here. So they are more resistant than enveloped virus, but more sensitive than others small and non-enveloped virus. So they are a good target for disinfection according to EPA. Thank you.